Dear students, today I am just going to deliver the topic fertilization in the plants, in angiosperm plants, how the fertilization takes place there. So, before that, we have just completed the development of male gametophyte, female gametophyte, the journey of the pollen grain from anther to the stigma of the plant. So now everything is all set for the fertilization, and as the fertilization will complete, the embryonic development of the plant will begin. So just beginning, I'm just going to begin with the very small, very easy concept you knows very well. In the previous chapter only we have just seen what is the meaning of fertilization. Fertilization is fusion of the both male and female gamete. Fertilization is the fusion of both male and female gamete. So so after fusion of male and female gamete, what will happen? They will just going to produce what? A diploid structure. That diploid structure is called as zygote. So what is the basic concept of the fertilization? The fusion of male and female gamete is going to produce a diploid structure. That diploid structure is called as zygote. So zygote is always diploid. We have a long discussion over there. And in case of plant, the important question is there, where the fertilization will occur. The site of fertilization in the case of plant is always the embryo sac. Embryo sac means to say that is a female gametophyte. What is embryo sac? Embryo sac is considered as the female gametophyte. Female gametophyte means the structure which bears the female gametes are eggs. So is it clear to you all where the fertilization occur? The fertilization occur inside the embryo sac or inside the female gametophyte. Now just take a look one by one ki what are the basic process or mechanism of the fertilization there in the plants. So first of all, the germination of the pollen grain, or we can say the growth of pollen tube. What is the mechanism of the fertilization? The first step of the fertilization there in the plant, first one is that germination of the pollen grain or the growth of pollen tube. So just see one by one, we are just going to we are just going to understand how the pollen grain are going to germinate there over. So first of all, as soon as the pollination gets complete, the pollen grain will travel the journey and they will land on the surface of stigma. And here the very first process is there to identify the particular type of pollen grain, whether the pollen grain is belonging to the same type of the flower or not. Means what? It is being checked by the stigma only whether the pollen grain is of the same type of flower or it is of the different type of the flower. So if it belongs to the different type of the flower, so it will not be able to germinate over there. So just take care, just listen it carefully ki we cannot say that ki every flower or every pollen grain can germinate on any of the flower. So how it is being made possible, how this is going to be justified, we knows earlier only. So first of all, what happened, the compatibility checking process is going to be taking place on the surface of stigma. We knows very well the tapetum of the pollen grain is releasing some of the recognition factor along with the pollen grain, and same type of the factors are present there on the surface of stigma also. So at that time, this is a completely genetic process. Genes are going to determine whether the pollen grain is of the same species or not. If the pollen grain is of the same species, of the same pollen grain, same type of the flower, then it will be allowed to get germinate. Otherwise there is no chance of pollination occurs. So just after identification of a particular type of pollen grain, now stigma is going to allow the pollen grain to undergo in the process of germination. So how the germination began? Actually we have just completed the process earlier also, but now I'm just going to revise it, revise the process. What happens ki as soon as the pollen grain is identified, now the pollen grain is allowed to absorb the moisture and the nutrient from the surface of stigma. What happens after identification? Now the pollen grains are allowed to absorb the nutrient and the moisture from the stigma. What are the nutrient? Basically the sugar, and these nutrients are absorbed by the pollen grains from the stigma. And we knows very well in the earlier topic only, in earlier lectures only, I have just made you clear कि by absorbing the moisture, by absorbing the nutrient, the cytoplasmic content of the microspore or pollen grain is getting increased, and due to the increased content of the cytoplasm is getting is going to exert a pressure on the intine, and intine is coming out in form form of a pollen tube from any of the germ pore. So is it clear? That prolongation, that extension of the intine is called as pollen tube. So this is what the basic process, what happened, basically identification, then absorption of moisture and nutrient. Due to the moisture and nutrient, the cytoplasmic content will increase, and finally the intine will be pushed outward in form of the pollen tube. And you knows very well, in the pollen tube, firstly the vegetative nucleus or tube nucleus will enter, and it will be followed by the generative nucleus. So what are the factors which are defining, what are the factors which are determining the process of germination or process of pollen tube growth thereon? So I'm just going to listing down one by one. The first of all, the first factor which is required for the germination, one that is sugar. As now we have seen, the sugar is being used as the nutrient for the purpose of the pollen tube growth. The second one is very important. The second one, the second factor is called as boron. Always it is being used there in artificial culture medium. Also we are using boron so that it could produce or it could enhance the pollen tube formation. So boron, how it enhances the pollen tube growth or how it enhances the enlarging the pollen tube, it actually useful in the transport of sugar, and it reduces the chances of pollen grain bursting. Are you getting my point? How it is useful? It is useful in transporting the sugar from top to the bottom, and it helps in reducing the chances of bursting of the pollen grain. Next one, calcium. Some enzymes are also there which are required for the pollen tube growth. The enzymes are present very much there in the pollen grain only or in the microspore only. The enzymes cellulase, you know very well, to digest the cellulose cell wall. Pectinase, because pectocellulose cell wall was present there. Callase, to digest the callose present in between the microspore. These all enzymes are present in the microspores are there. And to increase the cell growth and to increase the elongation, the auxin and gibberellin hormones are also required. The temperature, the optimal temperature range for the process of pollen tube growth, that is 20 degrees C to 30 degrees centigrade. This is the optimal range of the temperature. One very important point, the growth of pollen tube is always apical, that already we have discussed in the previous lectures. The growth of pollen tube is always apical and it is chemotropic. Why this is chemotropic? We have already discussed when we were studying about the 7-celled 8-nucleated structure of the embryo sac. The tapetum is having some specified structure called as the filiform apparatus. Filiform apparatus are being known to release some of the chemicals or the hormones which attracts the pollen tube toward them and guide the pollen tube to enter in the embryo sac. So that is why, because this is completely influenced by the chemical and the chemicals are released by the synergids. So this is what, this is completely a chemotropic or chemotactic movement. So as soon as the pollen grains germinate on the surface of the stigma, they will enter in the style. So means what? The growth of pollen tube always occurs inside the style. Where the pollen tube growth will be there, it will occur inside the style. So the style is different in the structure in different type of the plants or different type of the flower. Like that, there are three different type of styles are present. First one is called as solid style. The style is completely solid inside, so that is basically present in the dicot. Here it is very much clear, the solid tissues are being present there inside the style. So this is very much difficult to penetrate those tissues and growth or elongation of the pollen tube. So for the purpose of making the passage to the growth of pollen tube, what happens? The pectinase enzyme is working there to dissolve all those content whichever is present there inside the solid style. Are you getting my point? To increase the length of pollen tube, to provide a passage of growth of pollen tube inside the style, in the hard tissues or in the solid style, pectinase is working to dissolve the tissues thereon. Now next one, the second type is hollow style. So this is present there in the monocot, and I don't think there is any kind of the problem. Easily the pollen tube grow inside. And the very important feature of that kind of the style is that the complete cavity of the style is lined with the glandular tissues. Means they are just going to release the definite type or different type of the enzymes, materials, nutrients and other secretions thereon. The semi-solid type of the style is also reported, that semi-solid type of style is reported there only in the family Cactaceae. So till now it is very much clear to you all what happens. I'm just going to revise once again the entire content whatever I just delivered till now. The process of fertilization is basically fusion of male and female gamete which forms the zygote, where it occurs inside the female gametophyte or the embryo sac. The first step of the germination, first step of the fertilization is the germination of the pollen grain or growth of pollen tube. What it does, how it occurs: first of all, identification of the particular type of pollen grain, then absorption of the sugar nutrient, and then the growth of pollen tube from the intine only, that will be facilitated by the sugar, boron, some calcium ion, some enzyme like cellulase, pectinase, calase, etc., and hormones like auxin and gibberellins. And the growth is always apical. Since the growth is moving downward in the top region, the vegetative nucleus is present always in the apical position, and this is also referred as the chemotactic mode of the development, because the growth of the pollen tube is guided or regulated by the chemicals released by the synergid cells present in the embryo sac. So pollen tube grows inside the style that we have seen. Three different type of the styles are there: solid style, hollow style, and the semi-solid style. Now till now we have seen the pollen tube is the structure which is conveying the gamete, means the pollen tube is the structure which is conveying or carrying the male gamete to the site of fertilization. That is what the pollen tube is, the structure which is carrying the male gamete to the site of fertilization. Means what? This is the structure by which the fertilization is being completed. So that is what the process of fertilization in the plants is also referred as siphonogamy. Gamy, you know very well, the fusion. Siphon means tube. So the fusion with the help of siphon or the pollen tube is called as siphonogamy. So here what happened? What is the meaning of siphonogamy? Fertilization is completed with the help of pollen tube. If you have to define the siphonogamy, so siphonogamy means what? The process of fertilization which gets completed with the help of pollen tube is called as siphonogamy. Pollen tube, why this is important or how it is important, how it is helping in the process of fertilization? The pollen tube is going to convey or help in the transportation of the male gamete to the site of fertilization. Here you have to take care, we must understand the pollen tubes are based on the number of pollen tube growths. The siphonogamy may be of two types. First one is called as monosiphonous. Are you getting what I am saying? Based on the number of pollen tube growths inside the style, the siphonogamy may be of two types. First one is called as monosiphonous. Monosiphonous, single, mono, siphonous, siphon. So if only one pollen tube, if single pollen tube is going to develop, that is called as what? Monosiphonous. This is very much common in the angiospermic plant, most common in the angiospermic plant, and the best example of that, capsula, that we are always understanding in the complete embryonic development, because the complete embryonic development was studied there in the capsula only. Capsula model is the ideal model for the embryonic development. Now next type of the siphonogamy is called as polysiphonous. Polysiphonous, if more than one pollen tube is being grown there, so that is called as polysiphonous. So if single pollen tube is there, so that is called as monosiphonous. If more than one pollen tube is there, then it is called as polysiphonous. But remember, just take care, just just keep in your mind, in case of polysiphonous also, there may be a chance the pollen tube may be 1, 2, maybe 2, 4, 10, 12, whatever the number is there, but only one pollen tube is able to mature and will reach to the site of fertilization. Are you getting my point? How many numbers of pollen tube are developed, that doesn't matter, but only one is going to mature and can reach to the embryo sac, means to the site of fertilization. So is it clear to you all? So where we can report the presence of the polysiphonous condition? In the family Malvaceae and the Cucurbitaceae. In Malvaceae and Cucurbitaceae, the polysiphonous is there, and monosiphonous is present in most of the angiospermic plant, even there in capsula also. So till now we have just completed the complete development of pollen tube, means the pollen tube is now ready to enter there inside the embryo sac. inside the female gametophyte. So let's understand, understand ki how the pollen tube is entering inside the ovule, or how the pollen tube is entering inside the embryo sac. So whenever the pollen tube is going to enter inside the ovule, so every time it is being facilitated by the obturator, which is the part of ovary. The entry of the pollen tube inside the ovule is guided by the obturator, which is the part of ovary. So inside ovary, the obturator is there, which is guiding the passage or pathway of the pollen tube inside the ovule. So what are the passage by which the pollen tube enters inside the ovule? Just see, the first passage or first way of entering is called as, first way of entering the pollen tube inside the ovule is called as sporogamy. Poro means what? Spore means pores, aperture, opening. So we knows very well only one opening or only one aperture is present there in the pollen tube, sorry, in the embryo sac or ovule, that is micropyle. So when pollen tube enters inside the ovule with the help of micropyle or through the micropylar region, when pollen tube enters in the ovule towards or through the micropylar region, then it is called as sporogamy. This is very much common, that means it is the case of most of the angiosperm along with the capsula. Second way to enter inside the ovule is the chalazial opening. So second way is to enter inside the ovule is through the chalaza only. So that is the called as chalazogamy, when the pollen tube entering in the ovule by the mean of chalaza. So the process is called as chalazogamy. The chalazogamy is reported there in the casuarina, batula and juglans. Third one, mesogamy. The third process or third way to enter the pollen tube into the ovule is mesogamy. When the pollen tube taking some other ways or alternative ways except chalaza and the micropyle. So two alternative ways are there. Either the pollen tube may enters with the help of funicle, or it may enters with the help of integument. Are you getting my point? Usually porogamy with the help of micropyle or through from the micropyle, chalazogamy from chalaza only, and mesogamy when these two portion or these two pathways are left and any other alternative pathway is being taken to enter inside the ovule. So if the funicle is there, so example of our funicle is pistachia, and if integument is being taken, so cucurbita is the good example for the mesogamy. Two very important point that I am just going to make it clear here. That is only the important point for the NEET examination also. In Brassica oleracea, both two types of the two methods of entering inside the ovule is observed. That is, porogamy means through the micropylar end or micropylar region, and mesogamy means either from funicle or from integument. So in Brassica oleracea there are three different ways, either from funicle or from the integument or from the micropylar region only. So till now we have just made it clear that the pollen tube is entering inside the ovule either from the micropylar end or from the chalazial end, or it may took the way of or passage of the funicle or of the integument also. But remember, my dear child, I am just going to make a point very much clear. The pollen tube may enter from any of the may enter inside the ovule from any of the part, whether from chalaza, whether from micropyle, whether from the integument or funicle, but whenever it has to enter inside the embryo sac, so it is forced to take the pathway of micropyle. It is forced to take the pathway of micropyle. Are you getting my point? Pollen tube may enters from any of the passages, from chalaza, from micropyle, from funicle or from integument inside the ovule, but whenever it has to enter inside the embryo sac, so this is required, it should have to enter from the end of micropyle. If the question is there ki why this is necessary, so just remember the micropylar end is having the egg apparatus. So as soon as the pollen tube will enter through the micropylar end, so the male gamete will have to interact with the egg, so that is the easier one to get fertilize the egg. So now it is clear how the pollen tube is entering inside the ovule. Now we have to see ki how the pollen tube is entering inside the embryo sac. Till now we have seen how the pollen tube is entering inside the ovule. Now this is the time to understand how the pollen tube is going to enter inside the embryo sac. So just try to understand. Inside the embryo sac, always it will take the micropylar, and since the egg apparatus is present therein, so always it will have to take the micropylar pathway. So what is the way in to enter inside the embryo sac? The first one, it can enter in between the egg cell and the synergid cell. It between egg cell and the synergid cell means it will bisect the synergid cell and present. It will take the passage of in between the synergid cell and the egg cell. It can bisect the two synergid cell. Third one, in between the synergid cell and the wall of embryo sac. The fourth one, the bisecting the one synergid cell. So are you getting my point? And here in all the passage, what is common? The synergid is common. So every time the pollen tube has to penetrate the synergid, means whenever the pollen tube is entering inside the embryo sac, which structure has to be penetrated? Synergid. And you knows very well, synergid is the structure which is attracting the pollen tube toward them by the releasing some of the chemicals. They are having filiform apparatus to guide the movement of pollen tube also. So is it clear? So always the pollen tube has to enter inside the embryo sac from micropylar end and with the help of synergid. So two reasons, if the question is there, why the micropylar end is required to enter, why the pollen tube is always required to enter from the micropylar end? Presence of synergid, because chemotropic movement is there and the synergid is releasing some of the chemicals thereon. And the second reason is that the egg apparatus is lying there, so this is very much easier process to get fertilized therein. So next one, so it is clear now. What happens, as soon as the pollen grains as soon as the pollen tube enters inside the embryo sac, the pollen tube get burst out and it is going to release or discharge all its content inside the synergid cell. Are you getting my point? As soon as the pollen tube enters inside the embryo sac, it get burst and it discharge whatever the content present there inside the pollen tube to the degenerating, because it is going to enter inside the synergid, and synergid is also getting degenerated, so it will discharge all the content into the degenerating synergid cells. So degenerating synergid cell mein what the content it will release? Remember, what is the content present there in the pollen tube? First of all, one vegetative nucleus or the tube nucleus and two male gametes were present there in the pollen tube. So all three will be released inside the degenerating synergid cells, and along with some extent of the cytoplasm present over there. One very important factor has been observed in the synergid cells at the time of discharging of these materials into the synergid cell. X bodies are been reported. What happens at the time of discharging these material of the pollen tube or degenerating pollen tube into the degenerating synergid? The synergid has an appearance of some darkly stained oval bodies thereon. The synergid is having some darkly stained oval bodies therein. Where these darkly stained oval bodies are there, they are present there in the degenerated synergid or degenerating synergid, and these bodies are being asked there in the NEET examination ki what are the remarcation are there. So the synergid is having two degenerating bodies, two oval shaped degenerating bodies. These are darkly stained and these bodies are called as X bodies. So these X bodies are the remains of degenerating nucleus of the synergid and the vegetative nucleus. Are you getting my point? What is there? What is this? This is the degenerating synergid nucleus and the vegetative nucleus. So now it is clear to you all what is X bodies. X bodies are the darkly stained bodies or darkly stained structure present in the degenerating synergid, and they are the representation of the degenerating synergid nucleus and the vegetative nucleus. So it is clear till now कि whenever the pollen tube has to enter in the ovule, so either from micropyle porogamy or from the chalejogamy or from the funicle or integument, that is mesogamy, but whenever it has to enter inside the embryo sac, it has to take the route of micropyle only. The when it enters inside the embryo sac, what it does between synergid cells and the egg cell, between two synergid, between synergid and the wall of embryo sac, or bisecting any one of the synergid. Most common way is that one only. So in each and every content, in each and every pathway, the synergid is common because synergid is providing the chemicals to attract the pollen tube there only. Is it clear? Two reasons I have just made you clear ki why the pollen tube always enters from the direction of the micropyle. The first one is that one, the chemical is released from the synergid only, and second one is that ki the fertilization is taking place with the fusion of male and female gamete, and female gamete, that egg, is lying on the micropylar end only. And we have seen that X bodies are present there on the degenerating synergid, and these X bodies are what? Degenerating synergid nucleus and the degenerating vegetative nucleus. Okay, so two steps we have completed of the fertilization. First of all, the pollen germination and growth of pollen tube. The second step, how the pollen tube enters in the site of fertilization, that is the embryo sac. And when it enters in the site of fertilization, so what happen? The syngamy and the triple fusion. So third step of the fertilization is syngamy and triple fusion. What all this we are just going to see. What is syngamy? So first of all, syngamy, you knows very well, syn together, gammas marriage. So the fusion of male and female gamete is called as syngamy. This is actually the fertilization. So syngamy is the fusion of male and female gamete. What is triple fusion? That we'll understand later on in the topic only. So first time the fertilization in the plant was reported by the Strasburger in 1884 in the monotropa. That is very important question for the NEET examination. Many time it has been asked who has reported the fertilization for the first time. So it was Strasburger who has reported the fertilization in monotropa in 1884. What is happening there during the syngamy? Very easy, because the challenges are there to reach the male gamete to the site of fertilization, and we have completed all the challenges. So fusion of one male gamete, listen, fusion of one male gamete to the egg cell is called as the syngamy. Fusion of one male gamete to the egg cell is called as syngamy. Remember, at the time of development of male gametophyte, I just put a question there, why two male gametes are there in the angiospermic plant. So we'll understand here only why those two male gametes are there and what is the use of second male gamete. Because the first male gamete, the one male gamete, which one is coming further, which one coming the first after the tube nucleus, is being used as the fertilizing event, or used as the male gamete to fertilize the egg cell and produce the zygote, that is a diploid structure. This is also a true fertilization because here the zygote is formed. So whenever or wherever a diploid cell zygote is formed, that is called as the true fertilization. And one important term I have just written, that is generative fertilization. Why this is called as generative fertilization? Because the male gametes are being produced from the division of generative nucleus and the gametes are being fused. So that is what the generative fertilization. Fertilization is completed by the cell produced from the cells produced from the generative nucleus. Okay, so now the next one is double fertilization and triple fusion. Here the second male gamete will be used. What is the importance of that male gamete? You will just understand. The double fertilization and triple fusion, I will just explain each and every word. You will be able to understand what is the actual meaning of that one. Very important question we are dealing here. What is double fertilization and what is triple fusion? The double fertilization was first time studied by Navasin. Very important question. Just go through it. Just go through, cram it. It was studied by Navasin in 1898 in Lilium and Fritillaria species. Who studied the double fertilization first time? Navasin in 1898 in the Lilium and Fritillaria species. What happened in the double fertilization? The first male gamete, two male gametes were produced. The first male gamete was fused with the female gamete and produced the zygote, that is the first fertilization. Second male gamete, which remains unused till now, that second male gamete is going to join the polar nuclei or the dikaryon. Are you getting my point? The second male gamete is going to fuse with the polar nuclei or the dikaryon which is lying in between. Why this is so? Because as soon as the first gamete is being used, the second gamete is discharged in the embryo sac and it is traveling ahead. So whenever it is traveling ahead, the first cell which has to counteract, that is what the polar nuclei. So polar nuclei, it has to join to the polar nuclei. That is what the second time fertilization. So two time fertilization is there. What we are just going to understand. Polar nuclei, you knows very well, the condition of the polar nuclei is n plus n2, n condition is there, and another male gamete is going to fuse there. So that is what another n. So n plus n plus n. So here three nuclei are going to fuse there. In case of second time fertilization, what happened? The three nuclei, two nuclei from the polar nuclei are dikaryon and one nuclei from the male gamete, second male gamete is going to fuse. So that is what triple fusion. Three nuclei are getting fused together. That process is called as the triple fusion. The triple fusion is going to really just going to form a kind of triploid structure, n plus n plus n. So that is what triploid, not the diploid. So triple fusion is going to produce a triploid structure. That triploid structure is called as PEN, primary endosperm nucleus. That is triploid in nature. What it does, the triple fusion is going to lead the development or production of the triploid nucleus, that is called as PEN, primary endosperm nucleus. The primary endosperm nucleus is further and the finally is going to convert in form of the endosperm. How it will form the endosperm we will discuss separately in the next lecture. So primary endosperm nucleus is going to develop in form of the endosperm. And remember my dear child, the endosperm is the source of reserved energy, the source of energy inside the embryo, which is going to provide the energy for the embryonic development. So this is the reason the second male gamete was produced and is used or consumed in the process of formation of endosperm. This is the importance of the second male gamete. Now what is the basic concept? How many nuclei are involved there in the double fertilization? If you can count there, how many nuclei are involved there in the double fertilization? So if you can count number of nuclei is involved there in the double fertilization, so may it be 3 or 4 or 5? It is 5. Why this is 5? Because two nuclei from the synkaryon, two nuclei from the male gamete and one nuclei from the egg cell. So egg cell is equal to egg cell 1, 2 male gamete 2 and 2 from the synkaryon or the polar nuclei. And how many gametes are being used there in the double fertilization? 3 gametes, 2 male gamete and 1 female gamete is required. So in the conclusion only, what is double fertilization? So double fertilization, why this is so? Syngamy plus triple fusion is equal to double fertilization. Why this is referred as double fertilization? Because fertilization occurs twice, 2 times fertilization occurs. First time it forms the egg, second time it forms the primary endosperm nucleus. First time it forms the zygote, and second time it is going to form the primary endosperm nucleus, which is later on going to form the endosperm. And very important point that I am just going to discuss here, the double fertilization is the property of angiospermic plant only. The double fertilization is the property of angiospermic plant only. This is not found there in the gymnosperm plant. So it is clear to you all what is syngamy and the double fertilization. Syngamy, double fertilization, triple fusion. Syngamy and triple fusion combinedly called as the double fertilization. What happened? The first male gamete is fused to the female gamete or the egg cell to produce the zygote, that is the true fertilization or the first fertilization. Because remember, whenever the zygote is being produced, that time it is considered as referred as the true fertilization. Second time, the second male gamete is moving ahead and will fuse to the dikaryon or the polar nuclei and will form a triploid structure, that is called the primary endosperm nucleus. So that is not the true fertilization, but the fusion of male and female gamete occurs. So 2 times fertilization occurs in case of the angiospermic plant. That is why this is called as what? Double fertilization. So what is the meaning of double fertilization? First time syngamy, second time the triple fusion. So syngamy plus triple fusion is equal to double fertilization. Importance: zygote is produced from the syngamy and triple fusion is going to produce the endosperm, which is going to provide nutrition for the embryonic development. So is it clear to you all till now? Here I'm just going to show some of the diagram here only, like that the condition of monosiphonous and the polysiphonous. The first one, growth of pollen tube is there, so siphonogamy is there, and siphonogamy is of two type, monosiphonous and the polysiphonous. And here the pollen grain fall on the surface of a stigma and single pollen tube is going to generate, and that pollen tube is reaching toward the final stage only, that means to the embryo sac or ovule only. In the next diagram, what is there, many of the pollen tube started developing thereon, because great number of the pollen grains are fall on the surface of stigma, and each and every pollen grain, if they are being checked their compatibility, so they are just going to try, they are just trying to develop their own pollen tube, and only one pollen tube is able to grow completely and will reach to the final site of fertilization, that is embryo sac. And can't you think that that is also a kind of natural selection? Means what, the pollen grains are trying to survive themselves in form of growing the maximum pollen, growing the mature pollen tube. If they are able to grow the mature pollen tube, means their gametes are being shifted into the female gamete and their seeds will be produced. And if this is not possible, so their existence will get over there only. Now this is what the entry of the pollen tube inside the ovule. The first one, that is sporogamy, means if you have remember, this is the micropyle. So from the micropylar end, if it is going to enter, that is sporogamy. Second one, the pollen tube is entering from the chalaza, so that is what called as chalazogamy. The third one, either it is going to enter from the funicle or through the integument. Either it is going to enter from the side of funicle or through the integument, that is called as mesogamy. So chalazogamy from the chalaza, porogamy from the micropylar end, and mesogamy from the funicle or integument. But here in each and every case you are able to observe, even there in mesogamy, it is entering inside the embryo sac, through the micropyle. In chalazogamy also it is entering inside the embryo sac through the micropyle, and porogamy is itself entering from the micropylar end only. What happens there in the fertilization event? After entering inside, the pollen tube enters by either bisecting here in between the synergid cell and the pollen sac wall, in between the synergid only, in between bisecting this one. So like that, every time the synergid is involved, as soon as it enters inside the embryo sac, it will blast, it will burst and will release the content thereon. So first of all, the first male gamete is going to release and will get fertilized with the help of egg and form the zygote. The second one, which is behind that, is going to move ahead and will join to the polar nuclei and form a triploid structure, that is called as PEN. So that is this is what the first time fertilization, and this is what the second time fertilization. This is first fertilization or true fertilization, going to lead to the formation of zygote, and second time this is going to form the triploid structure, that is called as the PEN. PEN just going to form the endosperm. So two times fertilization, first fertilization, second fertilization, so twice fertilization. That is why this is called as what, double fertilization. And why it is called as triple fusion? Because already the two N plus N was there and one another N is there from the male gamete, so that is why this is called as what, triple fusion. So double fertilization, triple fusion, just take care, very important condition is there, very important structure is there. And remember the ploidy level also, the male gamete, female gamete, they are having what, haploid number of the gametes are haploid nature, the zygote is diploid, endosperm is triploid. So this is all clear to you all. So till now I just clear each and everything regarding fertilization. Now very important portion I have to start, that is what, till now we have seen the double fertilization or the triple fusion is going to produce a structure that is called as PEN. What is the fate of this PEN and what is the importance of this PEN, I will just deliver in the next lecture. So till the time, thank you.